Jeffrey Keller is one of those really broad-based scientists who has not only worked on basic biology of aging, but has taken it all the way to the clinic. He's doing clinical trials, he's doing basic research. I would describe him as a biochemist, a systems biologist, a disease biologist, an aging biologist. He started his career, or he got his PhD, I should say, in 1999 at the University of Kentucky. And in the 13 years since, he's managed to write 157 publications in really top journals. Ryan, take note. You better go home and write another 10 papers tonight. He has served as an editor on numerous journals. He's written 3 books that I know of, and possibly has a few more on the side. He served on multiple NIH panels, so I discovered a few minutes ago, and has an incredible CV that has spanned whole systems to the aging neurodegeneration component too. His work has looked at high-fat diets and diabetes, has looked at proteasome and oxidative damage, and has looked at obesity and Alzheimer's disease. And I'm not going to waste much more time of his in this introduction, just to say how very pleased I am you came and how lovely it was to meet you yesterday. Thanks a lot.
Thank you. So, wow, he wasn't kidding. He was going to activate this thing. So I want to thank Shelley for having me here, and thank the warm welcome I've gotten from everybody here at the Barshop in UTHSC, a lot of letters. Want to thank Carl in particular for spending so much time, really appreciate it. So I hope today to have a talk of, that really sort of spawns a lot of discussion. So what Shelley outlined, broad can be, it's nice to finally get to a stage in your career where broad does not mean unfocused. So that's kind of nice. I have, you know, it's really interesting. I think I finally found where I'm supposed to be. You know, I was lucky enough to get funding for yeast work, for primary culture work, for rodent work, for human work, to do clinical trials, NIH funded, and then do pharma. And so that full spectrum has got me to a place where it's sort of a unique perspective with which I look at things. And I think that hopefully that's going to spawn a lot of discussion here. I sort of organized this in 3 ways. The first is to talk about aging, obesity. It's so nice to be at a place where you don't have to convince them that aging is an important topic, so I'm not going to spend that much time on it. Obesity, I'm going to show you a few surprises. And with brain aging, I think that we're really in the middle of the largest, one of the most important human experiments in the history of mankind. We're at a time where just in the United States, you have 10,000 people a day turning 65, and 66% of Americans are overweight and obese. And this merger and the synergy between these 2 things has a tremendous impact for the field of aging, and I'll be telling you some of the thoughts. I'm going to go into a little bit of whether or not there really is something such as Alzheimer's and some of the controversies and ways we're tackling the study of Alzheimer's disease in our trials and basic research. And then this is a real important one. I'm going to talk a little bit about using a registry to study human aging. So I think that the Barshop in particular could benefit from some registry types of studies. And I'll show you what we're doing in Louisiana and hopefully build some collaboration or at least ideas on what you could do here. So obesity, aging, and brain aging, obesity, and brain aging. We always see graphs like this where life expectancy is increasing. And of course, that's, you know, the average lifespan that's increasing in the United States. Whenever you look at areas, specific states, we know that there are a lot of states where average lifespan is decreasing. And one side note is, you know, it's kind of interesting, one of the biggest predictors for Alzheimer's disease is area code. And you can, these belts that run through, you know, we can outline these areas as the stroke belt, the cancer belt, you know, whatever belt you want to name. It's sort of right here in these ripe areas, and we always call Louisiana the belt buckle. So you have this, you know, this thing going on, and of course there's lots of causes and lots of factors going on, but one of the more interesting ones is this graph. So this is where they really find the losers. So what this is areas that are green have actually the lowest incidence. They have the highest lifespan. Well, I'm sorry, they have the highest leisure time activity, the lowest obesity, and the lowest diabetes. And then the areas that are tan are the rest of us. So you know, that you have this spectrum. And this is just to start the conversation of how things like physical activity, things like obesity, are very, very important on, and will have an impact on aging and overall health and how we study aging. And there we go again with the belt buckle. This is one that's surprising to a lot of people. So these are the different age groups in the United States. The dark purple represents in 1978-79, and this represents from 2004. And what they are is the percentage in each of these age groups that are obese. And so what you see is, you know, we always have a lot of focus here, you know, on children and young adults, middle adults, this huge increase in the amount that are obese. But it's also to be noted that in the 75 range is also one of the most dramatic areas where we have increased obesity. And it's really, it's unprecedented. And so it's undoubtedly that this level of obesity will change the way people age in ways that we can't foresee, ways we better be studying. And it's also going to affect how we manage age-related diseases. And I'll just give you a few examples. So 25 years ago, there was no such thing as type 2 diabetes in children. Didn't happen. Now, we have a huge increase in the number of kids that are diabetic. You say, so what? You know, that's okay. That's really not going to have an impact on overall health. When you think about type 2 diabetes for decades and what that's going to do to the trajectory of diseases in that kid, you also have issues of things like management. There's starting to be papers coming out now on how early should you give statins to children. So, you know, all of these issues, it's going to change the way that we arrive at our elder years, and it's going to affect how our elder years actually progress. And I'll give you another example. So you say something like diabetes. So if you're convinced that diabetes is important to brain health, which I hope to have you leaving here convinced that it's very important, one of the factors is that 27% of the people who have diabetes don't even know it. Okay? So that raises all sorts of issues about screening in the elderly and looking for these complications of obesity. Obesity affects everything. It affects multiple systems. These are the biggies right here: diabetes, dyslipidemia, and hypertension. We know that each of these is important to brain health. And while there's great work being done on obesity, there's very little work being done on the effects of obesity on these different tissues. And that's one of the things I'll be talking about today. That's an area that we really need a lot of work in, and that's an area where someplace like the Barshop could really excel. A little bit about obesity. When I shared with some of you at dinner and my different conversations, we moved to the Pennington because we saw how important obesity and diabetes were going to be to the brain and neuroscience research. And we were at a place that's one of the best for neurology and neuropathology. And we knew very little about the endocrine system and studying obesity and what high-fat diets do you use and how do you do this kind of research. So we made a decision to move to Pennington, where they do that very well, and apply our neurology and neuropath experience on there. And what we learned is that we probably shouldn't have gotten into obesity to begin with. It's so complicated. And I'll just give you a couple of examples here of how complicated obesity is. Most people think about fat as this big inert tissue. It's actually the largest endocrine tissue in most Americans, okay? You start there. And fat isn't fat. You know, you hear this apple and pear shape. Well, a large amount of visceral adiposity is linked to disease. You name the disease. It's linked to visceral adiposity, not subcutaneous fat. And here's an example. You know, if you look down here, so you can see that within the visceral cavity, you have all of this adipose tissue. In mice, you have male mice, epididymal, retroperitoneal. You have these specific depots of fat. It's biochemically distinct, as I'll show you later on, than subcutaneous fat. Subcutaneous fat is that fat that's found near the skin, also in your hips and buttocks. And that fat is actually protective in many, many instances to metabolic disease. And I'll show you some examples of where we do a lot of transplants to study it. But suffice it to say, fat is not fat. It depends on where it's at. And visceral fat, bad. Subcutaneous fat, definitely not as bad, possibly good. This is a graph showing, so this is the highest level of visceral adiposity. This is the lowest. And this shows survival curves in the elderly. And so the amount of visceral fat that you have is very important to overall morbidity, okay, and mortality. If I did body weight, the curves don't look like this. If I did adiposity, the curves look kind of like this, but not as good. Visceral adiposity is really the key. So when we study aging and when we study the effects of fat on it, it's really, what we're really interested in is specific depots and their role in regulating aging. Let's take something as simple as losing weight. So I just told you all of this about fat and how horrible it is. Should old people who are overweight or obese lose weight? What does the room say? Yeah. No, we have some no's. Do we have any yeses? But the thing is, nobody knows. You take something, it tells you how wild this field is. Something as simple as that, we don't have the answer to. So I'll give you an example. There's a wonderful gentleman, Villarreal, over in New Mexico. He's doing great work, large studies looking at weight loss in elderly individuals. He can have people lose weight in a variety of paradigms where they control food, exercise, etc. Here they are losing their weight. You say, great, that's wonderful, they're losing weight. Well, they also lose bone density. And ask any of our geriatric researchers, and they'll tell you that bone density loss, we're not real big fans of, okay? And that happens across the board whenever you lose weight as an elderly individual. How quickly does it happen?
Are we talking within 10% body weight loss, or is it within 2 months? Is there a relationship?
There's a very good relationship. It tends to be you tend to lose a little less, for every % body weight that you lose a little bit less than that of your bone density, depending on the bone, if you look at overall bone density. And so that's a very big concern. Frailty, I don't have to tell you, one of the weight loss in late life is one of the biggest risk factors for death. So that's another factor. And there's big studies going on right now to try to understand, should we have weight loss in the elderly? Should we not? This is the point where I always have to remember to say, Dr. Keller is not saying everybody should get fat and live a long, healthy life. I'm not saying that. Of course, it has problems for your knees. It has problems for sleep. It has problems for a whole host of things. That's not what I'm saying. But what I'm saying is it's much more complex than lose weight. It's much more complex than that, which leads to one of the most fascinating things. This will blow your mind. So we have people at Pennington who do a lot of work with gastric bypass, gastric sleeves, Roux-en-Y, all of those surgery procedures. So you've all heard about them. What you probably don't know is how they actually work. So you take all of these procedures, and what ends up happening is people typically lose about 15% body weight. Okay? That's great. That's a lot. They're still morbidly obese. They don't do that. Well, now they're starting to do them. They're considering doing them on children. So, I mean, they're starting to go. They keep pushing it back. The bariatric surgeons and the insurance companies are fighting, and they keep trying to push it earlier and earlier and earlier. But that's another subject. But suffice it to say that you still have to be morbidly obese to get one of these procedures done. So you lose 15% and you weigh 350 pounds. You still weigh 300 pounds. Okay? The amazing thing is they have a longer lifespan. Those people live longer. Not only that, they live healthier. Those people have lower incidence of metabolic disease, dramatic improvements. And what's also amazing is you can give those people gastric bypass surgery, and within days their insulin resistance is improved. They haven't even started losing weight.
Question.
Yeah.
They live longer than obese people, or they live longer than general population?
Those papers are by a guy named Lars Solstrom, and the largest studies that they have on that are in Sweden. And so what they do is that they look at people. Sweden has a nationalized insurance and health registry and all of that. So what they're able to do there is they're able to take people who are age matched. They can match for everything, and they can look at the lifespan of people who are morbidly obese versus those that had the surgery. And they're able to do that with great accuracy, unlike here in America, because everybody there has the same insurance and such. So, and that's where he's a wonderful man. That's Lars Solstrom. So there's, so that's where that is. So if nothing else, I want you, if you hate everything else that I'm about to show you, leave here knowing that the study of obesity and the brain is going to be it's a very fascinating field, it's particularly for the field of aging, and it's wide open. And a place like Barshop, who does all of the work with health span and et cetera, I think it's just really well poised to start doing some really important work in that area that needs to be done. So, what are the effects of obesity on the brain? I'm going to get on my soapbox for just a second. So, almost nothing known. There's almost nothing known on this. We've published a large number of papers. There's a few other groups that have done work. And in general, what all of us have shown is that, yeah, you can make the brain get a little bit worse, and I'm going to show you some of that. You can alter important neurochemical pathways, and I'll show you some of that, in cognitive disturbances and some interactions with aging, or with Alzheimer's disease. But one of the things that's important is that the field really, so people are not consistent with how long, what strains of mice that they use. Does this sound familiar? You know, that's the strains. The diets that people use. So some people will use saturated, unsaturated, 45%, 40%. Some people will use with hypercholesteremia model. Some people won't. Some people use a chow as a control. Some people use the actual control diet, which you should use. Pair feeding isn't really done. And so it's really hard to understand this literature. It's really hard to understand what's going on. And I'll give you an example. If you do a study with a high-fat diet and compare it to chow, the chow diet does not have the same source of protein. It does not have the same source of fat, the same source of carbohydrates. We really don't understand what's going on with that comparison. We do know that they become fat, they become fatter, but we really don't understand, is it the fat in the diet? And it gets to those wonderful questions that Arlen and everybody else used with the caloric restriction stuff. You know, if you do a pair feeding study, which you need to do, you're actually restricting the diet accessibility. So you're studying something else there too. So diet studies are really complicated is what I'm getting at. But we need to start having sort of a standard in the field for how we do the diets and look at their effects on different tissues. So I'll show you a couple of examples. So this is one of our recent studies, and we give animals, in this case, it's a 45% lard diet, and control diet is the control. So it's they're isocaloric, and what they miss is the amount of fat in the control diet. And so we can make the animals put on weight. It's almost exclusively due to increase in fat. In this case, I'll show you a couple of examples with oxidative stress. So in these, yep.
When you said they're isocaloric, so have you compensated for the energy content of the fat with protein or with carbohydrates?
Carbohydrates. But that's what, but so you have all of these choices with which to make, so maybe towards the end we can argue on what's the optimal control. But I think pair feeding is actually probably the best to do. But this is what we did, and so in this instance, so with oxidative stress, we did electron paramagnetic resonance measures. So you take a little piece of brain, you can incubate it with different label, and you can use different antioxidants to measure specific species as well as different spin labels. And what we find is in the brain that total ROS, superoxide, peroxynitrite increased by the diets. Voila, okay? But one of the real interesting things that happens is that if you look at how each of these is related to cognitive function also declined in these same animals, and you look at how the reactive oxygen species relate to cognitive function, body weight, and adiposity, what you find is that basically the ROS are not related to any of the measures of any of the cognitive measures. They're just not. So we do. So we do a lot of different. There's a lot of different ways to do it. So I know Don Ingram was here last weekend or whatever, and one of the things that we do with Don is we use his stone maze, which is a wonderful way to do it, where you the animal does not have to swim. It's just it has an aversion to the water that it's in. It has to make 14 choices in this maze, and so you can get around poor vision. You can get around poor mobility. You can get around a lot of those things and count errors and latency. We also do a fair bit of work with fear conditioning. That's another one where you can get around the fact that they're less mobile, poor eyesight, etc. So fear conditioning is one of our big ones, as well as the 14-unit stone maze. And when you look at this, there's something that we're going to pursue a little bit more, and I'll just highlight that here. So here's body weight compared to each of the ROS, and here's adiposity compared to each of the ROS. And what's really kind of interesting is I'm not so interested in total ROS. That's pretty similar between the groups. But this one in particular is kind of interesting. So adiposity has a 0.92, whereas body weight has a 0.69. And so there's a couple of things that we're gonna pursue this smell a little bit more. This seems kind of ripe to us. So understanding the we think that if we did visceral adiposity on this, that it would be through the roof versus subcutaneous. So we're going to start pursuing this a little bit more. Another one that I'll give you is cholinergic signaling. So we do a lot of work. Alzheimer's disease is a disease for which we have no disease-modifying medication. The medication that's given the most increases something called acetylcholine in the brain. Acetylcholine loss and cholinergic signaling loss is very correlated with disease progression. And so we did a study where we looked at the effects of high-fat diets, these same guys, and you look to see if we affected the enzymes that are involved in synthesizing acetylcholine and the enzymes that are involved in degrading acetylcholine. And yeah.
But in this experiment, you did not find that this paradigm affected cognitive function, is that correct?
No, we did have a change in cognitive function.
But it doesn't correlate.
It doesn't. It doesn't. And that's also a very interesting tidbit. That is true. So we absolutely did. So if you look here, this is with control diet, high-fat diet, hippocampal formation. We see dramatic decreases with choline acetyltransferase, as well as increases in acetylcholinesterase activity. We've also done studies where we give the animals donepezil, so Aricept, which inhibits this enzyme from breaking down acetylcholine. And you can improve many, but not all, cognitive measures, and we're still trying to figure that out. Didn't want to show the data on that. But it does look like there is an involvement with acetylcholine loss and obesity effects on brain function. And so, so we're very interested in that. There's also a definite tie-in there with acetylcholine and Alzheimer's disease, neurotransmitter signaling. So we're going to pursue that a little bit more. Something else that I'll be talking about later on is this disease called cerebral amyloid angiopathy. And I'll give you a little more on that in a moment. It's a disease that's caused by amyloid formation within the blood vessels of the brain. And we looked at whether or not high-fat diets could alter the levels of cerebral amyloid angiopathy. It's a very robust effect within the vessels of the brain, getting this amyloid deposition, and it's also altering blood flow, and we've just started those studies looking at that. You do see, so here's a case, wild type, control diet, high-fat diet, control diet, high-fat diet, looking at synaptic markers in GFAP. And we do see a change within both the synapsin as well as an increased exacerbation of the GFAP from this CAA mouse model. So here we have high-fat diets on the brain affecting multiple levels of pathology, whether it be just a normal mouse or a mouse with CAA pathology. But let's talk about mice and obesity studies. So mice are wonderful. They're very, very wonderful for studying obesity. There's no question about it. That's how we found leptin. Studying the endocrine functions of adipose tissue, very important with mice. Adipose biology, the causes, especially the genetics of obesity, very useful. But they're not so useful or have limitations on studying the ability of obesity to modulate different tissues. And some of these, you know, obese humans, most obese humans develop a metabolic syndrome. It's not the same in everybody, but it has multiple components. Very few obese people are just diabetic. There's a component of dyslipidemia, hypertension, et cetera. And so they really fail to replicate most aspects of metabolic syndrome. It's a very, very, very modest increase in insulin resistance in response to a high-fat diet. I'm talking about diet-induced obesity. I'm not talking about DB and OB mice. This is with the diets. Modest increase in hyperlipidemia, modest in hypertension. It's virtually impossible to give a mouse atherosclerosis. And really, you can't study the use of statins and other things. You give a mouse statins, it has little to no effects on the lipids. It's still very anti-inflammatory and other things. But they have real limitations in understanding obesity in humans. And so we have to recognize that. Doesn't mean that we can't use them, but these are all very real things. Be interesting to see if the non-inbred strains, you know, can overcome some of these difficulties, as well as novel models like the mole rat that is closer to the guinea pig. Since guinea pigs are wonderful, they have a wonderful lipid profile very similar to humans. And so that's very exciting that we may be able to tackle some of these problems. What I've chosen to do is follow Ron Kahn, so another Kentuckian. He heads the Joslin at Harvard, and Ron is an amazing guy. He can get a cell paper with 10 negative figures and 1 positive figure. I haven't figured out how he does it yet, but Ron is one of those people that I've followed since I started graduate school, and always been amazed at. And now I have a chance to do some of the similar work. And so what we've started to do in mice is to understand visceral adiposity versus subcutaneous fat. How do they affect the health of the brain and other types of factors? Ron's done a really wonderful job showing visceral fat is not the same as subcutaneous fat, and he developed a transplantation model. I do all of the surgeries myself. I was trained by the vet there, and so this is my chance to escape the office, and so I'm very, very dedicated to these experiments. I got this grant funded, and so what we did was we looked at the effects of visceral fat on CAA, cerebral amyloid angiopathy. And so what we do is you take an animal 2 months or 8 months old, we give it 1 gram of visceral adiposity. So I increase the visceral adiposity in the mouse, the donor mouse. I use a green fluorescent protein, so I use the donor mice from GFP positive, so I can follow the transplant's health, which I won't bother showing you. But, and actually the next slide's kind of messed up. It's one of the things that got messed up in this talk, but it's a model that works very well. You can transplant this fat, it stays in there, stays healthy for a year. I think we've gone up to 18 months. And it has a very big advantage because you can transplant from adiponectin negative mice or leptin overexpressing mice. You can do all sorts of really cool combinations, which Ron uses to study fat and metabolism. We would use them to study fat signaling to the brain. That's our goal. And so, yeah.
Do you control the diet of those mice that you're taking the transplants from? Is there any way in which
You can
You've
do.
manipulated that.
So
with
Ron
fat pads.
has done a lot of those experiments where you donate fat from obese animals versus lean animals and all of that. We haven't gotten that far with it since I do all the surgeries. So what we have done is, but I've done a lot of them, and I'll also show you lipectomies. We're also studying the effect of lipectomies. So our first goal is visceral adiposity. What if we make it go up? How does it affect the brain? What if we make it go down? How does it affect the brain?
You're just making the donor mice, and your hands are just regular mice. You just take the visceral.
The only modification is that it's GFP positive mice so that we can follow it.
And
Yep.
what strain is it?
They're both C57. And so, so what we ended up, and note that it's a very modest, it's 1 gram. So you're talking about a 3% change, okay, an overall adiposity. It's very low. And what ends up happening is you lose your slide when you come to San Antonio. So what you would have seen on this figure right here would have been that the body weight is the same. There's almost no change in body weight. It's very similar. It's not statistically different. If you look at the adiposity, it's significantly increased. And this slide was taken, and I got if anybody wants to see it, I'll show you on the printout. But there's a sustained elevation of adiposity, but not body weight. And then if you look here, it shifted my slides around. But with glucose tolerance test, have a larger area under the curve when you give the transplant. Very modest, but it's significant. And then the fasting glucose levels are also elevated. And so this is 12 weeks following the surgery. So, but we have done it for up to, I want to say 18 months, but at least 12. But I think I've got a few of them that are still around that have had it for 18 months. And so it's a really great model that you can then start doing all sorts of really cool, playing with which adipokines are most important, looking at the effects of different depots on the brain. And so what do we see with the brain? Well, this top figure is with the electron paramagnetic resonance. SH refers to the sham animal. TR refers to the transplant. So we can increase total cerebral oxidative ROS as well as peroxynitrite in the cerebral cortex. With the stone maze, we can increase the number of errors as well as latency in these animals, so an impairment in cognitive function. We can increase our glial reactivity, alter synaptic markers. So we're very comfortable that this is a model that's having significant effects on the brain, and we're going to be pursuing this. What if we add more fat? What if we take the fat out? There's all sorts of combinations with which you can do. But it allows you to get away from all of those nasty things about the diet-induced obesity. Here we're studying what does visceral adiposity do to the brain. So then this is with the CAA. So if you take the sham and the transplant animal, we get a lot more vacuole. And this is a young animal. This animal is, what is 2 months plus 5 months? This is a 5-month-old animal. So usually with this mouse model, it takes 12 months to get this level of pathology. So we're getting higher levels of this vacuolarization of the brain. And these are thioflavin S positive deposits of the amyloid. And so I think I have, it's always wonderful to have collaborators. So one of my good friends up in Kentucky has started doing experiments with the scanning electron microscopy. I don't know if y'all know about this, but it's an old school technology that's coming back again. What is it in science? Every 7 years stuff comes back. So this is an area that's coming back. What it allows you to do is you can perfuse a rosin that you have to actually smuggle into the country from Germany. It's actually, you're not allowed to have it here, and it's small. They ignore it once it's here, but the deal is it's not supposed to be here. So, but anyway, you smuggle this rosin in, you perfuse your animals with it, and it perfuses them all the way down to the microvessels of the brain. And then what you can do is you can add proteases and digest all of the rest of the brain away, and what you're left with is the matrix of the vasculature of the brain. Then you can take an electron microscope and you can go in there and look at the microvessels and other vessels of the brain, look at changes in arborization. And what we were looking for was the microaneurysms and blood-brain barrier breaches. And what we found was here's the control animal, and you see these control means they're still APP mouse. This is not a healthy mouse. So this is a mouse that's going to go on to develop vascular disease. But at 5 months, we see very few of these anuritic bodies. And whenever we do the transplant, we find significant numbers, and you can see them here. They're all over here. There are areas where you get these anuritic bodies, and we're repeating the studies because you can actually have an anuritic emboli that come out, or you can have the breaches that occur, and they both look very similar, and we're repeating these studies to figure out which one it is. But suffice it to say, we can affect microvascular pathology in these mice with the visceral adiposity. This is collagen 4 and IBA1 staining for microglia, and what we find is that we have degenerating blood vessels of the brain, but that's not surprising in these mice. Like I told you, they develop CAA. But we also see increased number of the phagocytic microglia around the blood vessels of the brain when we do the transplant. And so we're very interested in understanding this pathology, and again, very relevant to human disease. This is the lipectomy work. So I did all of these surgeries. Yay! And yeah. Have not tried doing it.
Look at the vasculature.
Right. Well, this is vasculature of the brain, so not vasculature of the adipose tissue. Although they've started doing that at Pennington since we're doing this technology, but that's with the brain. No, we haven't looked at brown adipose tissue. Of course, that's only going to be in the subcutaneous fat of specific depots. But that is a question that I get asked whenever I'm at the Pennington and give these talks. So brown adipose tissue, of course, is biochemically distinct from white adipose tissue and is more metabolically active. So, so with the lipectomy, these are 3 months following surgery. So we have a decrease in body weight. It's almost exclusively fat, just a little bit of muscle loss, but primarily fat that's lost in these mice. And we're going to be doing studies to look at that effect on the brain. Do not, but we know that it'll be changed. So it's 100% that anytime you affect obesity, whether manually, because Ron Kahn's done some of that, looking at the microflora in response to some of these transplants, and it definitely occurs. But the nice thing about a transplant is you can put in adiponectin positive, negative, leptin positive, negative, and start teasing apart what part of adiposity is important to mediate that signal. So now we're going to switch gears a little bit. So I'm going to do a little bit of clinical. We're going to start getting into the clinical realm. We're going to, well, no, actually, I'm going to throw a little mouse in here too. So we haven't gone all the way clinical yet. But we're going to talk a little bit about Alzheimer's disease. So one of the things with my background, my postdoc was with a wonderful man named William Mark Sperry, one of the best neuropathologists in the country. And with Bill, God love him, one of the things that I started picking up on early was we had a lot of bad controls. So we had people that died who, when we went to consensus conference to look at their clinical record versus their pathological record, there were people who died with pathological Alzheimer's disease who were cognitively intact. And Bill would say they were bad controls. You know, that's just a bad control. Or he would say they didn't live long enough. And we'd say, Billy lived 89 years old. How long does he have to live? And it's just a bad control. And so that persisted for decades in the field and kind of got us into the mess where we are today. But suffice it to say that there is, if you look for people with pure AD, people with just the hallmark pathology of Alzheimer's disease, who have that cognitive decline consistent with Alzheimer's disease, who lack other neurological abnormalities, there you have a very, very, very small subset. The others small, less than 5%. The other thing that you note is that there are people who have those pathologies who are cognitively intact. So that starts us to start thinking about what pathologies cause the disease, how does all this fit together? And one of the things that really stands out to me is that, you know, whenever I see my people come into the clinic as part of our studies is that nobody has just Alzheimer's disease. It's a disease of the elderly. Okay? We don't have people that come in who just have Alzheimer's disease. They've got diabetes. They have other issues that are going on. And these things are things that we need to quit ignoring and to start understanding how they influence. Because the one thing that's consistent about Alzheimer's disease is that it's different in everybody who has it. And different, I mean by the rates of progression, the response to medications, the behavioral abnormalities, all of the pathology that we see. It's different. It's different in everyone. It's consistent with the syndrome. I'll give you another example. So my father's dying of cancer, and I started getting more interested in cancers because of that. And what's really amazing whenever you look at cancers is 25 years ago, all cancers were the same. Cancer was cancer. It didn't matter whether it was lung, pancreatic, hepatic. Cancer was cancer. Everybody got Red Death, and everybody either made it on the other side or didn't make it on the other side. And people started to understand that each of the types of cancers had their own thing with regards to microenvironment, their genetic profile. All of these things are different in the different types of cancers. Look where we're at today with survival curves for people with different types of cancer. And it's because we recognize that the causes are different, the early detection is different, the way you manage and treat is different. And that's where we're going with Alzheimer's syndromes. And I'll give you a couple of examples. So, another troubling figure that we see over and over again in the field, the only part of this figure that I agree with is that late-stage dementia and aging can be clearly separated. There's no doubt that we're very good at telling late-stage Alzheimer's disease from age-matched controls. We're very good at that. The further you go down this thing, the more problems that we have, and it's exponential. And you take something like mild cognitive impairment. What are we talking about? Are we talking about amnestic MCI, non-amnestic MCI, mixed MCI? What are we talking about? Because amnestic MCI, primarily memory, is what leads more often to a Alzheimer's disease-related dementia. But it's not all. We have people that go back and forth. We have people that go back and forth here. Most of the people that we see in our clinic are actually a mixed MCI. What the heck are they? What is causing that? And whenever you go preclinical prodromal, watch out. The amount of variance that we see and what we need to look for, very poorly understood. We see figures like this all the time. So here we have our healthy brain, our Alzheimer's brain. Temporal gyri goes to hell. We see lots of pathology here in late-stage disease. Our hippocampal gyri gets degenerated. We get a larger ventricle, less overall brain mass. That's late-stage disease. We don't see these huge structural changes early in disease. We don't see that. So we have to start thinking about Alzheimer's disease as not being just one disease, but early on, the causes and what we should be looking for, we still don't really understand. This PET scan, they use this image in all sorts of, came from the Alzheimer's, both of these came from Alzheimer's Association. This one in particular shows you the trouble with the disease. So they're using this one to try to highlight our attention to this temporal loss right here. Well, the thing that stands out to me is this frontal loss. Okay? We have this huge loss within the frontal cortex here of brain activity. This is glucose utilization. This figure should scare the heck out of you. There's nothing specific about this at all. And this is an Alzheimer's disease brain. We see lots of pathology. In reality, Alzheimer's disease is a disease of systems. It's multiple systems within the brain. People differ in response to their medications. The Aricept, Namenda, Exelon, 25% of the people get a little bit of benefit for 9 months. Alzheimer's disease is the number 6 killer total, okay? It doesn't happen in people under the age of 50, okay? We have these people, and it's still the number 6 killer. It's the only one in the top 10 that has no disease-modifying medication. Nothing. We're still at we're barely above where we were 20 years ago. We're starting to make advances, but we recognize that the differences in response to medications, the behavioral issues that develop in people are different, the progression, the pathologies present, and a lot of this is because we have no treatment. We have no treatment, so clinically it's become a catch-all. There's not a lot of work done to really diversify what types of dementia people have. And again, largely because there's no disease-modifying medication. So here, look, if we want to look at it this way, we have Alzheimer's disease, we have all of these differences, and highly likely that the pathology that's present, particularly white matter hyperintensities, that's a really big one. I'll give you the pharma trial that we just started. So we're doing a big study with Merck. And we go to the meeting, and we're defining Alzheimer's disease, and we have all of these guidelines on what Alzheimer's disease is. So if someone has 3 lacunar infarcts or less, and they have another clinical profile that's consistent with Alzheimer's disease, then that's Alzheimer's disease. If they have 4 or more, now we're talking about a vascular disease, and that's no longer Alzheimer's. Okay? And that's the state of the art. That's just one example. That's state of the art. You'll be reading about it in the paper. That's like one of the big, it's the big BACE inhibitor study that's coming out. And, you know, that's troubling. And people have, you know, when they have Alzheimer's, they have frontal atrophy, Lewy bodies, microinfarcts, white matter hyperintensities, and the one that I'll be showing is the CAA, Cerebral Amyloid Angiopathy. We really think that's one of the most important ones to study. And they also have hypertension, atherosclerosis, diabetes, obesity, increasingly. And all of these things with genetics impact these profiles. And so Alzheimer's disease is going to end up being a variety of syndromes that we tackle differently depending on how your profile of these factors are. Yep.
Everybody's looking at mild cognitive impairment, I see now in testing, because the assumption is that by the time you have the problems with Alzheimer's, it's too late to treat it. But from what you just said is, you're talking about that you could have a whole range of things that you call mild cognitive impairment, or am I wrong?
Correct. And no, and the NIA guidelines on defining mild cognitive impairment, they highlight the importance of using longitudinal studies because you need to follow people over time because they've had a decline. MCI is not a functional decline. They still have functional. They're not. That's where the big shift happens between MCI and AD is this functional impairment. But they have this big range of things called, and they give you guidelines on 1 to 1.5 standard deviations beyond test, and you can still be within normal range. But they have this catch-all for clinical judgment. And so clinical judgment, and that varies. Clinical judgment varies on what MCI is. And some people go through the trouble to figure out and separate if it's amnestic versus multi-domain or what. It's very messy. And it's very important to tease out what you're studying whenever you do an MCI type of study. And we can talk more about that. But that's, yeah, it's very important. Cerebral amyloid angiopathy, it's present in almost all Alzheimer's disease cases. It's in almost all Down syndrome cases, and it's the second leading cause of intracerebral hemorrhage. It's a very, very important pathology. It is a vascular problem, and you get this. You can get double barreling at late stages of the disease. Leptomeningeal is primarily where you see it on the with the meninges, but importantly you get these cortical arterioles that develop increasing levels of amyloid within them, and what it does is it just causes a loss of the basement membrane. The vessel just starts to deteriorate, and what's really what's really bad about this disease is that we don't have a good model for it. Van Nostrand has a very, we use that model. The data that I was showing you earlier was from the Van Nostrand mouse. It is a mouse model that it has a Swedish, Dutch, and Icelandic mutation within the A beta region. So we got three mutations going. It's expressed by Thy1, so fairly high levels of expression throughout the brain, primarily within the vessels, but kind of throughout the brain. And the problem with it is that those mice don't develop type 2. This is all type 2 CAA. This is what affects the larger vessels of the brain. The Van Nostrand mouse develops capillary problems, so very, very, very small blood vessels. You really can't stage it because they're so dang small, okay? You get a little bit of breakdown, you can't, it goes from 0 to nothing. And so basically what happens, if you want to think about it, the Van Nostrand mouse kills the twigs. It doesn't do the branches in the trunk, which is really what CAA is. So when I moved to the Pennington, we have a really wonderful transgenic facility, and so I wanted to see if we could make a CAA mouse model. We've had very good luck making humanized mice at Kentucky, and they were very important to developing. We do a lot of work with A beta solubility and changes in the profile, oligomerization, PIB binding, and all of that stuff. And what we wanted to do was to try to see if we made a humanized CAA, make a human, take the mouse APP gene, make the mouse A beta region human A beta, and then put in one of the mutations. We put in just the Dutch. And my thinking was we would do this, so we made a knock-in. And that's what I just described to you, humanizing the mouse A beta region and putting in one Dutch mutation. And our thought was first that we could do this in a year, which was moronic. And I now know a lot more about making the dang knock-in mouse. Oh, my God. But anyway, 3 years later, we're there. And so our thought was, it's going to happen. You know, Van Nostrand's has 3 mutations. It has a pretty aggressive promoter. So our knock-in is going to take longer to develop, but it'll probably be a better model in the end. Okay? So one of the postdocs is edgy in the lab, you know, I got to get papers, you know, can I go on and look at some of the mice? And I'm like, they're just 3 months old, you're wasting your time, but go ahead, let's start to look at them. And so what she did was she sectioned some of the brains, stained them for thioflavin, and I about jumped out of my pants. So what happened is that we see beautiful thioflavin labeling of large vessels of the brain. So in this case, these are 20-micron vessels. Okay, see, this is 3 months. So they're developing pathology that's relevant to human. These are 200 micron. We're talking very moderate to large size vessels here labeled with the amyloid. We can start to see pathology developing in these guys. And we do collagen 4, inflammation. We're doing all of the things that we should. And suffice it to say, 3 to 6 months, we're already seeing moderate CAA in these mice. And so that's really cool for the field because that's telling us that it's not having more of these things that's important and having multiple mutations. It's expressing it in the right size vessel that's important. And that's significant enough to affect pathology. We also get cognitive decline in these guys, which I won't bother showing you. But we're very excited by this, and we think that it's going to really change the field because CAA unifies so many diseases, and the vasculature is such an important part of obesity-mediated effects on the brain. And so we're going to be focusing a lot of efforts on CAA and that work. Yep.
Any deposits in the parenchyma?
Any what?
In the parenchyma?
You do. We do get amyloid within the parenchyma. But we're more interested in the vessel, but you definitely do. And even in human CAA, you see it there. And so people study CAA because they think it's important in plaque development, because plaques are always dense with blood vessels. And some people actually think that a lot of the senile and neuritic plaques that we see in Alzheimer's may be mediated by microhemorrhages as well as A beta from vessels. So yeah, very, very interesting area. All right, so this is my last part, using a registry to study human aging. So we really don't understand brain aging. And as a matter of fact, we don't even know what it looks like. That's really bad. We really don't understand what is a successful brain and what is an unsuccessful aged brain. We really have a poor handle on that. I always use this example. My son loves Abraham Lincoln, and we go to Washington, D.C. all the time, and there's a presidential museum, and in there they have a plaster cast of Abraham Lincoln, old Abe. About 9 months before he was elected president, he had a plaster cast made. I guess he had big thoughts even then. And they used this for a lot of the statues and things that we see out in paintings, et cetera, made molds from this. About 3 months before he was assassinated, so 3 years and 9 months later, thereabouts, less than 4 years, he had another plaster cast made. This one. And so everybody in this room looks at this and says, Abe ain't looking too hot, okay? Abe, this does not look like, and I'm not going to bother you with chronological versus biological aging in this room, but suffice it to say, this is not 4 years of biological aging. This is many more. And all of us can look at the furrowing of the brow, the sunkenness of the cheeks, and we can see that. That's what we need. I call this my mom test. My mom still has no idea what I do. She doesn't really understand it. You know, like, what are you doing? And I always use this as an example. We need to understand we need to be able to tell the brain is aged this way. Unsuccessful brain aging. So that's what we need to look for. I started something called the Louisiana Aging Brain Study, LABrainS, the Lamar Dementia Study. They're modeled off of what happens at University of Kentucky and the other 29 Alzheimer's centers around the country. This is a registry with which we use to have people that are cognitively intact. We follow them with a variety of tests, which I'll show you. And then once they convert to inactive dementia, we switch them over to the Lamar Dementia Study, where we have interventions for them, pharmacological and otherwise. I moved to Pennington, and they told me, as I told Steve earlier, they said that you can move there and we'll stay out of your way. We'll stay out of your way. You can raise all the money. You can go. Baton Rouge has a gentleman who's pretty infamous. I don't know how many of you know him, but there was this famous preacher. His name's Jimmy Swaggart. He still lives in Baton Rouge, and they say that I get into my Swaggart moments. So they said that I could go and they'd stay out of my way, and so I raised several $1 million, and I go and speak in church basements, groups, anywhere that they would have me, and I grew this study. The goal was to have 200 people. I thought if I had 200 people and I could do really good cognitive measures on them and follow them over time, that would be a really good registry with which to build a program around. And ultimately, one day, my goal was to have a pharmaceutical trial and an NIH grant. That was the big goal. So 4.5 years later, so this is the model. People come in for LABrainS year after year, with 92% retention year to year. That's key. You have to have these people buy in. They have to be, I told Steve earlier, you need them to be scared. So our people come to see us because they're scared of Alzheimer's, they're scared of falls. We provide free screening to them, and then we add all kinds of stuff to it. But the idea is, we call it, how much do you want to learn about yourself? Okay? Swaggart here, right? So we call it, how much do you want to learn about yourself? And we encourage them, and they ultimately usually go all in. And so what happens is that we have people that remain stable, people that develop cognitive impairment but still normal range, people that develop a dementia, and they come over to our Lamar Dementia Study. The other thing is, it's very easy to see here, you can then start to understand what causes people to stay stable, what caused people to move here, what caused people to move here, or what caused people to stay here. Okay? Lots of possibilities here. This is what I think you need. This is very, very important. So again, the goal was to get 200 people. So where we're at now is I have 2,000 people total in my studies, all 60 and over. We have participants from 34 parishes. I've established offices in Alexandria. There we work with the School of Allied Health. We have nursing, so it gives our students, the students there access to geriatric nursing experience. In Shreveport, we work with the medical school there. And in Monroe, that's the one I'm most excited about and our most recent. There we work with the School of Pharmacy. And so there's all types of polypharmacy and other things we can do. It's very important to get buy-in from each of these sites. Baton Rouge is by far our best. About 1,200 of my participants are here, 300 at each of the other sites, thereabouts. We do cognition. We do a full UDS workup like all the Alzheimer's centers do. We do a fall risk, depression measures, short physical performance battery, measures of nutrition, sleep, polypharmacy, anthros, lifestyle. We have all of these as part of our fit. The big thing that we do, though, unlike anybody in the country or the world, is we put an all-in bet on activity. And I'm going to try to convince you why that's so important and how it leads to animal studies that y'all could do here like no place else. So we do accelerometry, pedometer measures, life space measures, and I'm getting into driving, and you'll see why we do driving. So all of these are used for registries for our different studies as well as our future studies. So if you think about it naively, I got into this thinking, okay, physical activity, brain, obvious connection. We need to increase physical activity and look at its effects on the brain. Turns out it goes both ways. It's very, very interesting, and you can't study physical activity without looking at falls. And you can't study any of this without understanding the environment and life space. Life space is that concept of how far you move from your bed every day. 0 is you never leave your bed. 120 is you leave the same city unassisted every day. Okay, so there's these ranges. Show you here. normal cognition, cognitive impairment, dementia. What happens is that your life space gets smaller and smaller. I bet it happens in rodents. And if you give them a large enough space. And what happens is that within normal cognition, it's not just your space that changes and declines, it's also the intensity. So when you study physical activity, it's very important to study quality, quantity, and performance. It's not enough to know how many steps someone took. You need to know how fast they were moving, whether or not it was moderate to vigorous, how much inactivity that they have. All of these things are important to understanding their life space. Ultimately, this leads to frailty. Okay, it's not just dementia, it's leading to frailty and falls. And so one of the big questions, you know, we know this happens. Is it a cause or a consequence? And can we intervene? We can't keep people losing weight. It's so hard. We can make people lose weight, but we can't keep it off. We can't make people exercise. It's impossible. People won't maintain a routine. Increasing physical activity and space, we can do. We can intervene, okay? And we're successful doing it. And you can use things like smartphones and other things to really make it happen. So that's what's really exciting here, is that you might be able to intervene and increase physical activity, things that are very manageable and in reach and have very significant effects on overall health. We do it a variety of ways. So again, quality, quantity, patterns, that's what's important. So short physical performance battery, distraction. So we look at gait. I have people walk up a 10-meter, we walk 10 meters here. I look at stride length, cadence, velocity. I do it under distracted and non-distracted conditions. Ask mom to walk down, look at each of these factors. Ask her to do it again and spell the word world backwards, okay? Or do serial sevens, and I'll show you what happens. So there's big differences with regards to distractibility with gait. Real world measures like steps, real world measures with accelerometers, quality and patterns, and I'll show you. You have to build databases. One of the big things I had to learn is this thing takes second-by-second measures. So I take second-by-second measures from 2,000 people for 7 days, okay? So you have to come up with good programs with which to analyze that, but we've done that. We've overcome some of these hurdles. Driving, ultimately, is one of the big life space things. It's key. And, you know, we know, for example, people that are at highest risk for falls are at highest risk for car accidents, okay? We know that. What we don't know is people that are at highest risk for distracted gait, how does that relate to distracted driving? We're going to figure that out. That's one of the areas that we're going. So just think about this, okay? So if I told you take 150 older people, range in age from 55 to 92, maybe 65 to 92, I can't remember. But you take these people, okay, that are older. They're non-frail, okay? They're non-demented. And we say, what's going to predict, what are we going to see the biggest age-related change? Is it going to be in moderate to vigorous activity? Is it going to be the amount of, you know, like big activity that they do that's going to be the difference? Is it the amount of inactivity that they have that's going to be the difference? Or is it just the number of movements? Forget about steps, just the number of movements that people make. Okay, be thinking in your head, like what's going to be the biggest difference with age? This is what you see. Moderate to vigorous physical activity, no difference with age. Mean inactivity, no difference with age. And this should scare the hell out of you. These are healthy people, okay? These are healthy elderly people. They sit almost all day. 1440 is the maximum, okay? So most people are sitting almost all day. And this is what you can get your local politicians involved. You can actually improve these things. These are very easy things to intervene on. So here we have a ceiling effect. That's the problem here. Moderate to vigorous. Almost everybody, almost all seniors have way less than an hour of any moderate to vigorous physical activity. Okay? You're talking about if people went to 60 minutes, the average person had 60 minutes of moderate to vigorous, it would change everything. Okay? It's very low. Mean activity goes like this. Okay? So we've got a significant age thing with things. Now think, what measures steps better in older people? A pedometer or an accelerometer? Get in your head. What's going to be the best? Accelerometer. It is an accelerometer. So you actually look here, so pedometer. If you used a pedometer in older people, because pedometers aren't made to measure steps in older people. Okay? They're made to measure steps in adults. They're really horrible at children too, by the way. But they're really bad at measuring it in older people. Here you would not have a significant difference with age. But you do it with an accelerometer, much more sensitive in the elderly population. Okay, now you say, all right, what if you look in this population with these in-lab measures, okay? You take something like that's standard out there, like a frailty scale, or you take something out there like a life space. I just told you life space measures are very important. We work with Dr. Allman at UAB on that. And then we take something like our just total movements that a person makes in a day, and that's what it measures. It measures movements, whether it's a step, whether it's the moving of a foot, anything. It measures movement. No significant, and these are healthy individuals, so there's no significant difference with age with life space, no significant difference with frailty. Movements, big difference. That tells us, this tells us that in-lab measures are important. These are great. Life space and frailty are great. Just like we're great at measuring late-stage Alzheimer's disease from control, we're great at that. Okay? People starting down that road with aging and age-related change, we suck. Okay? We really do. Free living measures are the key. We really need to do free living measures. Yeah.
Sorry, I'm not sure if I missed something. These are cross-sectional measurements, right?
These
so
are
it's
measurements
not like
of
you're
the
following...
same individual. No, this is one time. Yeah.
Right.
So cross-sectional with age.
But can you predict how well they age based on changes in life space, frailty, or...
That's our bet. No one's done it. So that's our bet. We're going to do it in 2,000 people. So here you have this. So the next figure is like one of the most important ones. So, and I have great psychometrists that work for me. I try to learn from everybody that I hire. And one of the things these psychometrists always tell me is that when you give someone a test and you're trying to understand like test and what it means and how it fits with disease, you have to always wonder about people's motivation. Okay? What motivates them? Why are they there? How is that interaction with them when you're giving the tests? And then you have to step back and just take a look at all of that, okay? And so what I'm going to show you next, I think, highlights the difficulties and importance of why, if we're going to make any advances in aging and in falls, dementia, and other areas, we're going to have to combine these two. We're going to have to combine in-lab measures and free living measures. What we have here is steps per minute. Okay? This top figure right here is undistracted gait. Okay? So these individuals, this is from one person. Okay? This is one person, 74-year-old man. He had a step of 110 steps per minute whenever we had him walk down the platform. Said, walk at your normal speed. Okay? He walked. I said, walk at your normal speed and spell the word world backwards or the word cheese backwards or whatever word that we gave him, okay? It's slower, okay? No question, okay? That happens to everybody. This is his accelerometry, breaking down the steps per minute throughout the day. He does not walk one single minute at his normal speed during the day. Does not happen. So, is this his normal walking speed? Or is in here his normal walking speed? It's in here. It's in here. The best test results you get from people are when they're not being, when they're doing their normal activity. That's where you really understand what's going on. The key is you can't just use this. You have to use this and this, because we know that the difference between these two is highly, highly, highly predictive of falls. And we know that it's highly predictive of MCI. Okay? So that's where we're going with it. I think that's a really good example that highlights what's going on. You can do this with your animals.
So would that really be maximum cadence? I mean, is that what you're really looking at?
No, this is just steps per minute. So you can look at peak. You can look like peak for 30 minutes. You can look at average throughout the day. You can look at the average over 5 days. You can look, I mean, that's the problem. We have to figure all of this out.
No, but I'm talking about the upper line there of undistracted walk.
That is the average of 2 walks. That's correct. Yeah. So if you say, I don't care about anything that you've just shown, I don't think that walking and walking speed's important, I think it's all a joke and you're just wasting your time. Some gentleman put together a wonderful, and I should have him cited here, and it might be near Barzillai too, so I really got to watch what I'm doing here. But they put together a composite of hundreds of thousands of people, studies of walking speed and mortality. Okay? How fast someone walks at a given age and how long they lived after that. Okay? And what they find is walking speed is highly, highly, highly predictive of when you're going to die. Not only that, it's predictive of morbidities that are going to happen before you die, and that shouldn't be surprising. So, for example, if you're 65 years old and you walk at 0.4 meters per second, you're going to have a much shorter life than someone who walks 1.2 meters per second. Okay? And these hold. The beautiful thing here is you can intervene. Okay? There's ways to improve these. Okay? Yeah.
Are there? Because there are 2 ways of reading this graph. One is what you say. One is maybe there is some disease that already started, and that's why they work slower. That's why they will die sooner. So they won't die because they walk slower. They will die because there's a disease that's making them walk slower.
And that's why they call it, how fast does the Grim Reaper walk? So that is absolutely true. And what we know is interventions which increase walking speed lower, or they expand this. So it's not just cross-sectional here. And so we need to have these, what I'm saying is we need to have these curves for people in the free living condition so that we can then communicate with them by their smartphone to increase from here to here. And it needs to be combined with their metabolic profile, their medical adherence, and all of these factors. And they're very, very intervenable, okay? Very, very, very applicable to that. So this is just what we do. So we have a group of local physicians we also work with. So we don't just get our demented patients from my group. We get them from a variety of folks. We work them up better than the local groups. And we have a lot of collaborations with physicians and scientists. The School of Pharmacy is key. I have a project on health improvement outcomes where we look at diabetes management in people with Alzheimer's disease. We're developing biotechnologies which are important with the smartphone. We think smartphone's really the key thing. And I'm going to get the place to be an ADNI site in the coming years. These are all key. We're going to keep growing our pharma and our non-pharma work that we do. And these are the goals. But I just want to give you one last one. So the biggest thing that I think this will probably end up being the biggest thing that I do. This right here, changing the face of AD. So I think that we're going to have some really good interventions and ways to help folks and decrease the incidence and effects of dementia in folks. I do believe that's going to happen. I think it's going to take more time than anybody is really appreciative of. There's still time out there. I work with these people. I work with their families. I see them. And it drives me crazy that we can't do anything to make a significant impact on their life. Right now, when someone develops Alzheimer's disease, it's a death sentence. People know that. People deal with that in different ways, but it is a death sentence. So we need a way to change the face of Alzheimer's disease from being a current death sentence to something that provides hope, significant ways of management of the disease, and in particular, reducing institutionalization. Once that person's lost to the institution, that's one, it's a burden on that's another place you get your politicians involved. It's a burden on the communities. Everybody knows that, and it's a way to increase the quality of life for that person as well as their family. There are 3 things that cause people to be institutionalized, and all of them can be improved. Caregiver burnout, falls, medication adherence. These suckers right here are what cause people to go they determine when people go into the institution. And so what we're doing is we're using our smartphones and we're developing interventions. One works with peer groups. We're working with peer-to-peer as well as one-on-one burnout intervention for the care provider. Falls we're doing with the smartphone, help use of the accelerometer within the smartphone, and surveillance of the home for fall risk. Medication adherence is a wonderful thing. We're working with a group to, the University of Kentucky folks are very good at having medical adherence programs for a variety of cancers, smoking, et We're working with them to develop ways to improve medical adherence within the demented population. Medical adherence is one of the hottest things it's one of those rare things as a scientist where you can get paid from both sides. What I mean by that is the NIH, the VA, all of these places are willing to pay money for grants to help people take the medicines we know work. Okay? 40% of people on hypertensive medications are non-adherent. They're on a medication, we know that it works, we can't make them take the damn thing. Okay? The pharmaceutical companies are willing to fund medical adherence because if people aren't eating their pills, they don't get refills. Okay? So it's one of those beautiful instances of where you can help a person by making them take their medications and have two funding streams. And so we're very interested in pursuing that. But this right here is what I'm focusing a lot of attention on, and it's something that I would like to roll out at multiple sites. So if y'all get along, that's one area I think that we can help improve here too. I want to thank my lab and collaborators for the work that we do. We do a lot of work with the vet school. I want to thank my collaborators at Pennington and around the country and the different people who do it. I do want to point out one thing. The IDRP is very small. People were, when I was talking with some of you all, they were saying like, we don't have millions of dollars to do what you do. I see 2,000 people a year, 2,000 people a year, and it cost me about $310,000 for the staffing at my different sites to run this. There are registries, they're invaluable. I get NIH grants, pharmaceutical grants. We've now gotten to a stage where we're self-sufficient. But people still continue to give. They give because they see, you know, people here don't want to have to go to Austin to get their access to Alzheimer's trials. People here don't want to have to go to Houston. They want to get treatment here. You build a registry like this, you can make that happen. And so with that, I'll close and open it up for discussion. I appreciate your time.